First of all, I'd like to thank all the organizers of this symposium, especially Dr. Marcia Stefanik, and for this fantastic opportunity to present and share some of our research, as well as for our moderator, PJ, and to Howard for the kind introduction. And I just want to re-emphasize that much of what I'm about to present today was really rooted in fundamental biology started by Howard over a decade ago, fundamental biology, basic science, and genetics and epigenomics. And I'm very lucky and feel very lucky to be able to present this work to you today. So as clinicians and researchers, we're all too familiar with this striking demographic reality. Autoimmune diseases disproportionately affect women. If we look at historical data, we see a staggering female predominance in diseases like lupus, Sjogren's syndrome, and scleroderma. Over 80 to 90% of patients are women. So why does this sex-biased immunity exist? For a long time, the focus has understandably been on hormones, which Dr. Sabra Klein will beautifully illustrate in the context of infectious diseases later this afternoon. We also know that X chromosome dosage plays a role because of the many important immune genes on the X chromosome, such as TLR7 and FOXP3. But today, I want to focus on a third pillar, a very special RNA. This RNA, as Howard alluded to, is called XIST, and it's present in XX, but not XY cells. Female XX cells undergo a process called X chromosome inactivation, orchestrated by this RNA called XIST, and it compacts the chromosome into what we now know as the bar body. As shown in elegant work by Si Chu and Howard Chang in 2015, XIST forms an enormous RNA protein superstructure. It physically tethers to over 100 associated proteins, which we call XIST-associated proteins, or XAPs, to achieve this massive silencing feat. Here is where things get interesting for rheumatologists and clinicians. When we look up at the proteins making this complex, it turns out to be a surprisingly rich source of known autoantigens. We see SSB, or LA protein, HNRNP, U1RNP, HMGB1. These are well-described autoantibody targets in systemic autoimmunity. So our hypothesis was that this XIST supramolecular complex, this ribonuclear protein particle, can be a female-specific trigger of autoimmunity. Now, to test this hypothesis, we utilized an incredible tool developed with Anton Wutz at ETH Zurich. This is an inducible XIST mouse model. We wanted to see what happens when we introduce XIST into a male immune system in a mouse model of female autoimmunity. For this, we used the Pristane model. In this model, female mice develop hallmarks of autoimmune pathology such as autoantibody production and organ damage and even death, while male mice do not. And we wanted to test the impact of XIST independent of its role in X chromosome inactivation. I skipped over this part in the prior slide, but we can induce XIST in male mice because this form of XIST does not inactivate the X chromosome. Of course, inactivating the X chromosome in male mice would be lethal, but because this version of XIST forms the XIST RNP but does not inactivate the X chromosome, we can actually study the impact of the presence of XIST independent of its role in X chromosome inactivation. So the results were striking. In a Pristane-induced model, expressing XIST in male mice not only induced autoantibodies, but it also conferred female-level autoimmune organ pathology. Critically, we proved this occurs independent of female sex hormones because we're expressing XIST in male mice, and also independent of the function of XIST in X chromosome inactivation. So this points to a massive paradigm shift because we're talking about XIST acting as a physical scaffold for these autoantigens, and by its nature as a single-stranded RNA, potentially as an adjuvant for immune stimulation as well. We also show that XIST induces autoantibodies to its own RNP in this mouse model, and when comparing with autoantibodies from human patients, because we know known autoantigens, as I alluded to earlier, we show significant overlap between the autoantibodies induced to XIST RNP and the autoantibodies found in lupus patients. But how does this translate to human patients? And this is where we, namely Howard, realized the field had a massive systematic blind spot in the way we screen for autoimmune diseases. The gold standard indirect immunofluorescence screening test for autoimmune diseases uses a cell line, namely the HEP2 cell line, that is male-derived. So it completely lacks the XIST RNA protein complex. Furthermore, it turns out that the HEP2 cell line was actually contaminated by HeLa cells very early on in its derivation, to the point where the HEP2 cell line as we know it now is actually genetically identical to the HeLa cell line. And the HeLa cell line, despite its female origin, also does not express XIST. So essentially, we're using a cell line that lacks this very important female-specific nuclear structure to screen and test for anti-nuclear autoantibodies. And in fact, if you dig through the annals of science, there are papers where people did indirect immunofluorescence using patient sera on human cells, and they could actually detect autoimmune seroreactivity against the Barr body. And in a talk later by Dr. Lundberg, we'll also hear about how we can scale this up using optically pooled screening to further test for anti-XIST or anti-Barr body autoantibodies. But before that, we developed a protein array because we wanted to further explore XIST-associated protein autoantibodies in patients. So we built an array with both known autoantigens as well as XIST-associated protein antigens. And we found novel autoantibodies to XIST RNP in patients. One of our questions that followed from this, though, is that it seemed that only a subset of XIST-associated proteins were good at eliciting an autoantibody response, and some were not. And the question was, why were certain XAPs, but not others, dominant autoantigens? And to this, again, we based this in basic RNA and structural knowledge that was gleaned by Howard as well as many others in the XIST field about how the XIST RNA is structured and how it binds to its protein partners. Because our hypothesis was that the structure of XIST and where the RNA binding proteins bound to on XIST could guide the immunogenicity of some of these proteins. So we overlaid patient autoantibody profiles from patients who had lupus, scleroderma, and dermatomyositis, along with general healthy population controls onto the structural map of XIST binding proteins. Basically, if a patient had an autoantibody or reactivity against one of the XIST-associated proteins, we could map that to where it bound on the XIST RNA. And we found that the immunogenicity was not evenly distributed along the XIST RNA, as we predicted. In fact, anti-XIST-associated reactivity was concentrated in discrete hotspots, primarily localized to what we call the A and F repeat domains. And surprisingly, many hotspot XIST-associated proteins showed marked enrichment of expression in neutrophils. And this was very exciting for us because for intracellular antigens, particularly nuclear RNA protein complexes, they need to be released into the extracellular space and exposed to the immune system to elicit an autoantibody response. And neutrophils undergo this programmed cellular activation process called netosis, where it actually expels nucleic acid, DNA, and protein material into the extracellular space in the form of neutrophil extracellular traps, or nets. And we found that indeed we can find hotspot proteins such as SPEN and RBM15 present in neutrophil extracellular traps. These net structures stretching 20 to 30 microns effectively display the XIST RNPs to the extracellular immune milieu. So we have a model in which a very abundant cell type, which has enriched expression of these XIST-associated proteins, can become activated, release their nuclear contents in the form of RNA protein complexes, and we also hypothesize and are actively studying how the nature of XIST as a single-stranded RNA can directly serve as an adjuvant through TLR7 signaling to activate the immune system as well. We're also actively looking at the upstream triggers of netosis as well, because obviously this is something that may not be happening spontaneously. There needs to be a trigger, so we're actively exploring that as well. And importantly, going back to the question of why this might be relevant in patients, Dr. Lori Chung in the following presentation will present some really interesting work on how we might harness this knowledge about novel anti-XIST autoantibodies as new biomarkers in disease. And I believe that this autoreactivity against nucleic acid binding proteins is just the tip of the iceberg. The XIST RNA protein complex is actually one of many examples of RNA protein complexes in autoimmunity, namely the centromere, spliceosome, I'm sure many others. And I think a structurally based rationalistic approach to looking at autoantibodies against these nucleic acid protein complexes can pave the way forward for developing new biomarkers, new prognosticators, and ways of stratifying disease, ultimately to lead to the better management and treatment of patients. So our dream is to identify these patients and then develop screening assays against anti-XIST autoantibodies, leading to the development of new molecular diagnoses and endotypes, and ultimately lead towards developing new therapies. This is also the basis of the U54 project that was pioneered by Howard and then shepherded across the finish line by PJ, where basically we're trying to realize these goals and really translate decades of basic science into better outcomes and better lives for patients. So I'd like to acknowledge basically all the people who made this work possible, many of whom are in the room today, especially our SCORE project leaders, especially Howard, PJ, Marcia, and of course our research team members, and all the patients and donors who participate in our studies, all our funding sources, especially the newly awarded U54 grant. And with that, I'm happy to take any questions.